Okay, thank you. Please let me know if you can't see or hear adequately. So I'm going to talk about AAA ATPases. AAA ATPase is a very large family of machines that burn ATP to do mechanical work in the cell. And there are many different clades of AAA ATPases that do a variety of different drive a variety of different biological pathways. I'm going to talk about just one of them that we're going to call the AAA unfoldases. These are hexameric, usually hexameric proteins that are thought to drive unfolding of protein substrates by translocating them through the central pore of their hexameric structure. And I'm going to focus primarily on one of these AAA ATPases. I will introduce and discuss other AAA unfoldases as we go along, but I'm going to focus primarily upon VPS4, which is the AAA unfoldase that drives ESCRT pathways. So, what are ESCRT pathways? There are many ESCRT pathways, and what they do is drive membrane topology changes. The most ancient of these is probably the one circled here, which is the final stages of cell division, the pinching of the membranes off that sort of completes cell division. And all of these ESCRT pathways culminate in green polymers of ESCRT4. And you can see there are lots of them illustrated here. Here's one that's fixing wounds that may occur in the plasma membrane. Neuronal pruning seems to be driven by an ESCRT pathway, and there are many others. We got into this because my colleague Wes, some years ago, discovered that HIV, and it turns out many other viruses, bud from cells by recruiting an ESCRT pathway. Our understanding of how this occurs derives from insights from many different labs, a few of which are shown here. And the concept, the consensus thinking about how this works is illustrated in this beautiful animation from Janet Iwasa. I hope this is projecting okay in real time for you. But what I hope you can see is that there's a blue viral genomic RNA that is assembling with thousands of copies of the multi-domain GAG protein, which has like a light yellow, orange, and brick red domains that forms the major structural elements of the virus particle. In blue and red, you can see the envelope protein, remarkably few of which end up studded in the budded virus particle. And at this point, upstream ESCRT factors, of which there are a large number, are recruiting ESCRT3 subunits, which are shown here in the sort of bluey green color, that are forming a filament that is binding to the membrane at the neck and stabilizing it, we believe, in a highly bent metastable configuration. In turn, the ESCRT3 polymer recruits VPS4, shown in here in pinky purple color, which is unfolding ESCRT3 subunits and thereby throwing strain into the lipid bilayer that resolves by fission. And then the VPS4 continues to unfold ESCRT3 subunits so that multiple cycles of the pathway can continue. So there are many things to consider here. And the first topic I want to discuss is, what does ESCRT3 look like and how does it interact with the membrane? And I want to emphasize now that the work I'm going to tell you over the next few minutes is not from my lab. It's the work of Adam and Wes's labs, and we and others are part of a P50 consortium that is collaborating to study questions of HIV host interaction. But all the work I'm going to tell you about for the next few minutes is from Adam and Wes's groups. And I think they have the best model for what ESCRT3 looks like. But before I show it to you, there's a very important caveat that I need to be very clear about. And that is the ESCRT3 that they are talking about. And there are multiple ESCRT3s because there are 12 different ESCRT3 genes in human cells, for example. So there are different flavors of ESCRT3. This particular ESCRT3 functions in budding from endosome tubules. and so this is this budding has the opposite sort of positive curvature topology to almost all of the other ESCRT driven pathways that we've been looking at so far, which bud with the opposite topology. Despite that very important caveat, I think that the insights from Adam and Wes's structure are quite important. And so the first of these insights was published a few years ago, and it's a structure of a polymer between IST1 and Chimp1b, the two ESCRT3 subunits involved in this process. And it's very beautiful, as you can imagine, and there are two rings. There's a ring of Chimp1b subunits in the middle in darker green, and a ring of IST1 subunits on the outside. These are both ESCRT3 subunits, and they have related amino acid sequences. And the structures are somewhat the same in that they're very helical, and helices correspond to the same parts of the amino acid sequence, but the way the helices are folded up is very different. And this is a real head scratcher as to exactly what's going on here. So that structure was interesting, and more recently, very recently, Adam and Wes's groups published in NSMB a few more structures, including one of what is essentially a superimposable Chimp1b IST1 tube, but now bound with lipid bilayer. This is actually a pretty high resolution structure, though we're just looking at it in an overview form here. Let me see if I can get a laser pointer going. Okay, here we have it. The thing which is different now is that we have, in gray, we have lipid head groups from one leaflet of the bilayer on the outside and also on the inside. And the circumference of the inner and the outer ring differs by about a factor of 3. And this means that the lipid bilayer is extraordinarily stressed. It must be a very metastable configuration. So you can envision conceptually that as ESCRT3 subunits are pulled away from the lipid bilayer to relieve it in this metastable configuration, the strain can resolve by fission. And that's a pretty hand-wavy sort of explanation, but that's our currently, I think, the most attractive way of thinking about how ESCRT3 may be functioning in membrane fission. Okay, let's go back to thinking about VPS4. And the ESCRT3 recruits VPS4, which then acts upon the ESCRT3 to unfold it. In this representation, I'm showing the VPS4 discharging ESCRT3 into the lumen of the virus particle. That probably is not actually what's going on, but it was a convenient place to put the VPS4 so you could see it in this representation. So please forgive that artistic license. And that, we believe, drives the membrane fission. And then the VPS4 continues, we think, to do essentially exactly the same thing on the remaining ESCRT3s to reset the system. So the way in which we think the VPS4 works is illustrated in another of Janet's beautiful animations. At the top here, you can see the N-terminal domain of an ESCRT3 segment. That's the helical thing that in either its open or closed conformation stabilizes the filamentous assembly. And projecting down from that towards the C terminus, we have a flexible region which culminates in an MIM sequence motif. And this MIM domain provides the initial recognition by binding to the N-terminal domain of VPS4, which is an MIT domain. And that's connected to the VPS4, the ATPase cassette, by a flexible linker. VPS4, biochemically, is only a very weak hexamer, but we envision that when it's brought to high local concentration, such as through this lawn of interactions, that it's then able to hexamerize around the substrate and then associate with the yellow cofactor protein to bind and burn ATP, to walk along the substrate, and in so doing, pull the substrate through the central pore and unfold it. And so that's our thinking for how VPS4 may destabilize ESCRT3 filaments. this model derives in large part from a cryo-EM structure that I'm going to tell you momentarily. But before I do that, I want to emphasize that we were working on this for a long time. And so our first publications on VPS4 structure were actually back in 2005. And they included an NMR structure and a crystal structure of different domains. And on the left-hand side of the N-terminus, we have the 3 helix bundle, which is the MIT domain, and then there's like a 40 residue linker that goes into the ATPase cassette. And ATPase cassettes are comprised of a large ATPase domain in orange and a small ATPase domain in yellow. The nucleotide binds at the primarily to the large ATPase domain, but with the base at the interface with the small ATPase domain. And then green on the far side, we have a beta domain, which is important for recruiting that yellow cofactor protein. 2 other features I want to mention about the structure are Pore loops 1 and Pore loop 2. These are sequence motifs that were identified by others some time ago as being important in this class of AAA ATPases for engagement with substrate. Okay, so even back then in the dark ages, we were trying very hard to visualize VPS4 in its active hexameric state, but we were unable to do so until we switched to using cryo-EM. And so what I'm going to focus on now is the cryo-EM structure of this region of the complex, the ATPase cassette and the yellow cofactor protein, and a bit of green peptide that corresponds to substrate. Before I show you that, I want to acknowledge the people who actually did the work. This is driven by Nicole, a former postdoc, Han, a current postdoc, Peter, who's an assistant professor in our department and is an expert at EM methodology, and the project is collaborative with Wes. So part of the reason why this project was so difficult or so slow, I think, is in part is because VPS4 is only a weak hexamer biochemically. And so perhaps in desperation, Nicole forced VPS4 to become a hexamer by expressing it as a fusion with HCP1. I should be clear that HCP1 is a bacterial protein that functionally has nothing to do with VPS4 whatsoever, but it is a stable hexamer and so forces VPS4 to be hexameric. To further induce VPS4 to adopt the right sort of hexameric structure, Nicole included that yellow cofactor protein, specifically the VSL domain of VTA1. And Han made a very important contribution by building on work from Phyllis Hansen's lab to identify a peptide sequence in the C-terminal region of an ESCRT3 subunit that binds with a relatively high affinity to VPS4. These days, I actually think this was a bit of a fluke, and that I'm not at all sure that this sequence has any particular functional significance, but it was very reassuring at the time to know that we were dealing with a peptide that binds with a relatively high affinity, about micromolar affinity. Nicole and Han also discovered that the kind of nucleotide that was included was important. And so they could not detect binding in the fluorescence anisotropy experiment using ATP or ADP or ATP gamma S or AMP PMP. They could only detect binding using ADP metal fluorides, such as ADP beryllium fluoride. It's interesting to speculate why that might be. It might be because ADP metal fluorides can look either like ATP or can look like ADP or can look like some sort of transition state or intermediate. It's also possible that ADP metal fluorides are simply a structurally particularly good mimic of ATP. In any event, it was necessary and it was included. Also included, as is commonly done, was a pinch of glutaraldehyde to stabilize the complex. So the structure was determined initially at 4.3 angstrom resolution, and I won't say much about that, and then subsequently at 3.2 angstrom resolution using the Titan Krios and a K2 detector at the New York Structural Biology Center. And I want to thank Bridget and Clint and Zenning for their assistance with that. You can see the map representative, i.e. best parts of the map shown on the right-hand side, and you can decide whether this is a good map or not. We'll take a look at the map a little more completely. My background is in X-ray crystallography, so 3.2 angstrom resolution is pretty bad for some things. On the other hand, there's clearly the right size density in the right place for different amino acids, and it's pretty easy to trace the polypeptide chain in this structure. So it's not a very precise structure, but I do believe it's accurate. So the map is quite clear over much of the structure. And you can see it on the left-hand side here. And on the right-hand side, the good parts of the map correspond to 5 of the subunits. The red, orange, yellow, green, and cyan subunits have good density. And as we shall see momentarily, they form a helix that spirals away from us in this orientation. What you may should also be able to see is the blue subunit, the 6th subunit, is pulled away a little bit from the center of the hexamer, and its contacts of the large domain region in the middle of the hexamer are relatively open with its neighbors, and it has rather poor density. And in fact, focus classification shows that this subunit is occupying multiple positions as if caught in the act of transitioning between one end, the lagging end of the helix here, and the growing end of the helix there. That's a concept that we will revisit shortly. Before then, I just want to point out that yellow cofactor protein, which here is colored blue and gray, I won't say, the only thing I want to say about this in the talk is it acts like it's a staple. It kind of staples adjacent VPS4 subunits to each other. This is architecturally quite curious because the cofactor protein is two-fold symmetric, and it uses two-fold symmetric interfaces to bind to adjacent subunits, but it binds different, the interfaces it makes are different in each case. That's rather an amusing symmetry mismatch, but that's all I'm going to say about it. I'm also going to say a few words about nucleotide. So the association between subunits and the helical arrangement is stabilized by binding of ATP at the interfaces. And for the sake of transparency, I'm going to show you what the density looks like. These interfaces here are quite tight and essentially identical to each other. The other two interfaces are much more open. As best we can tell, the first 3 of the interfaces is bound to ATP beryllium fluoride in the ATP-like state. The density at this site, although the structure is essentially identical, looks more like ADP than ATP. I don't want to be dogmatic about that because this is it's a limited precision structure, and negative groups can be with oxygens can be difficult to visualize, but in cryo-EM. But that's what it looks like. Okay, so let's take a look at this helical assembly that I've mentioned a few times now, because this is really fundamental to our thinking of how these enzymes work. We orient by looking at the side, and then we'll clarify by removing that transitioning subunit. And you can really see how it's a helix. And we'll clarify it further by removing most of the VPS4 to leave just the Pore loop 1 and Pore loop 2 residues from each of the 5 subunits that are in the helix. And you can see it really is a very regular assembly. And we can demonstrate this by drawing a line along the helix axis, and then spokes from that to the Pore loop 1 residues of each of the subunits. And when you do this, you can see that it's a right-handed helix that, as you go from one subunit to the next, rotates 60 degrees and translocates 6.3 angstroms. And this is a very special structure, symmetry in protein structure, because it's the symmetry of a beta strand. Beta strands are often drawn as being straight arrows. And of course, in real structures, they adopt a wide range of local conformations depending upon the particular environment. But if you just take a fully extended polypeptide and you relax it to its nearest low energy state, it'll form a canonical beta strand, which is a right-handed helix that every 2 amino acids rotates 60 degrees and translocates about 6.5 angstroms. So even at very, very low resolution, the first time we could see the structure, even before we could see the peptide, we had a what I think is an attractive hypothesis, which also turns out to be true. And that is that the substrate is going to bind as a beta strand. The enzyme has this groove that has the symmetry of a beta strand, and it's attractive to think that peptides could bind in this way because almost any amino acid sequence can form a beta strand. Symmetry also implies that each of the VPS4 subunits binds successive dipeptides of the substrate in the same way. This is also, I think, an important concept for us that turns out to be true. So just thinking at low resolution, we can illustrate the conceptual implications of this structure in terms of a possible model of translocation. And to do that, we've drawn an idealized substrate exactly along the helix axis in a beta strand conformation in green. And then we recognize that the VPS4 structure is asymmetric. The 6 different subunits are in a different state, as if they're in different states along a rotary reaction pathway. And so we can envision the movement in response to ATP binding hydrolysis as just being a linear morphing between these different states. And when we do that, you can see how the VPS4 appears to walk along the substrate, which of course is topologically equivalent to pulling the substrate through the central pore. I think it's helpful to look at this from the top view, so let's do that. You can see the substrate is here in the middle, positioned in an idealized way along the helix axis. And in pink, we have ATP, and in gray, we have ADP, and there's an empty subunit over here. So as we start this going, ATP binds and hydrolyzes around the ring. And as it does so, 1 or 2 subunits let go of the substrate whilst the others maintain a grip. Okay, so this is a conceptual view of how VPS4 and other AAA ATPases may translocate and unfold proteins. Let's take a look at the higher resolution structure. VPS4 here is in white, and in colors we have the Pore loop 1 residues from each of the 5 subunits that make the major contribution to binding of the substrate. And we can clarify it by removing the transitioning subunit and then swing to the side. And in cyan, we have a rod going along the helix axis, and the peptide is in green. It doesn't lie exactly along the helix axis, but it wraps tightly around it, and as envisioned, it's forming a beta strand configuration. So this symmetry alignment between the substrate and the enzyme implies that there's going to be just 2 types of amino acid binding site on the enzyme. There's going to be one type of binding site for the amino acids on the left-hand side of the beta strand, and a different sort of binding site for amino acids on the right-hand side of the beta strand. And so we call these the odd and the even binding sites, because we're looking at the odd ones here, and they're called odd because it's for residue 1, 3, 5, and what have you. So we'll look at the first of these, and it's an aspartate 1, and its side chain is sandwiched between tryptophan 1 in red of subunit 1 and the equivalent tryptophan from Pore loop 1 in orange of subunit 2. We're now looking at the next residue in this sequence as an isoleucine, a totally different kind of amino acid, but binding in the same way between equivalent tryptophans of adjacent subunits. Now we're looking at the next residue in the sequence, asparagine 5. Again, a totally different kind of amino acid, but binding in the same way in this equivalent pocket between equivalent tryptophans from Pore loop 1 of adjacent subunits, flanked on one side by a lysine side chain that's also from Pore loop 1. Now we're looking at valine 7, the last residue in the sequence, which is binding in the same manner. So those are the odd residue binding sites. We'll now look on the other side of the beta strand at the even residue binding sites. To do this, we'll paint on residues from Pore loop 2, because they also make some contribution to binding here. And the first of these residues is a glutamate 2, and it's between this methionine from Pore loop 1 and the equivalent methionine in orange from the next subunit. And it's a bit harder to see on this side, but the same principles are being applied. We have a valine now, valine 4, which is binding between equivalent methionines from Pore loop 1 of adjacent subunits. And the binding sites are flanked on one side now by residues from Pore loop 2. This is a lysine side chain is the next one, doing the same thing. Now we have a leucine residue 8 binding in the same manner. So I think this is quite pretty. What happens is that the enzyme sets up this, by symmetry, sets up these arrays of equivalent binding sites that go down two sides of the beta strand. And the binding sites are hydrophobic notches that are quite large, that therefore can accommodate any side chain and naturally have favorable interactions with hydrophobic side chains. We think that polar side chains and even charged side chains can be accommodated because the pore is quite exposed, and so any hydrogen bonding requirements can be met by water molecules. And I'm going to emphasize that in the next slide, in this surface representation. So as we tilt it, you can sort of see how the transitioning subunit, now in purple, has got relatively large gaps on either side with the neighboring subunits. But the main point to focus on is the central pore, which is open all the way through. And in green, you can see that there's the peptide bound, oops. Oh, the blue pointer, okay. In green, we have the peptide bound to one side of the groove, and it's very open to solvent. So water molecules can easily get in there to satisfy any hydrogen bonding requirements. So let's take a look sort of more closely at the proposed translocation mechanism. In white, we have the structure of VPS4. And in colors, we have the tryptophan and the methionine from pore loop 1 of each subunit to make the major contribution to substrate binding. And 5 of them in red, orange, yellow, green, and blue spiral around in a helix. And this helical assembly is stabilized by binding of ATP at the subunit interfaces. And they form this binding site, which is optimized for a canonical beta strand. The 6th subunit is way over here, very disengaged from interactions with the substrate. And so the idea is that binding of ATP at the subunit interfaces stabilizes this helical assembly that forms the canonical substrate binding group. And then hydrolysis occurs here. It occurs at the green active site at the interface with the blue subunit. And then phosphate release destabilizes that interface, which allows the blue subunit to move towards the transitioning position. And in doing so, the binding site opens up and ADP can be released and the subunit lets go of the substrate. As this is happening, the transitioning subunit, the purple subunit, has an open active site. And so because ATP is present in the cell at about 10 times the concentration of ADP, it can bind ATP, which makes it competent to stack on the top of the helix to bind the red subunit, and in doing so, engage with the next 2 residues of the substrate. So that's the concept. Let's animate it just by doing linear interpolations. Here we're moving the transitioning subunit between the range of positions that it is seen by focus classification. We now envision that it's binding ATP to join the growing end of the helix and engage with the next 2 residues of the substrate. And the blue subunit's following along behind. We're going to get hydrolysis here at the yellow active site that will release the green subunit and allow it to release its ADP and open up and rebind ATP and therefore join the growing end of the helix and the yellow subunits following along behind, and so on. So this is a very sequential concept. So it's, you know, I think our structure was the first of this type to be published, but only by a few days. And at almost the same time, a whole bunch of structures came out of other AAA unfoldases in complex with peptide. And this is an incomplete listing of them. I apologize to the groups that have been omitted here. It's getting to be quite a long list, but there are multiple structures of AAA unfoldases in complex with peptide or substrate mimics. And so let's superimpose them. This is VPS4. with the peptide in green. And we'll clarify just by showing the Pore loop 1 residues. Now we're going to superimpose a few of those other structures on the ATPase cassettes and display just the Pore loop 1 residues. And we'll clarify it now by removing the transitioning subunits, which could reasonably be in a whole range of different positions. And you can see these different structures are remarkably similar to each other. And for me, this is a powerful implication that they're likely to work in the same way, or at least in the same way. And one thing I should make clear at this point is that the overall concept behind the mechanism that we're proposing is not completely original. So some time ago, the more Joshua Tor's group published a crystal structure, a beautiful crystal structure of E1 helicase in complex with DNA, and proposed a concept. This is a different sort of AAA ATPase, but they proposed a conceptually equivalent sort of mechanism. And then very shortly after that, James Berger's group published crystal structures of Rho helicase in complex with RNA. This is an RNA translocase, and again, a different sort of AAA ATPase. But the mechanism proposed is conceptually very much like the one that we're proposing for the protein unfoldases. Okay, I want to shift gears a little bit now. I'm going to introduce you to a protein. You can see it's a very happy protein. And the reason it's a happy protein is because it's thinking about an interesting question. And the interesting question is, how do AAA unfoldases engage their substrate? Now, one obvious possibility is that you have a terminus that can just stick into the hole. That's quite possible. An idea that I introduced earlier is that maybe AAA ATPases can hexamerize around the substrate. I think that may happen in some cases, but there may be more to it than that. Can AAA unfoldases translocate loops and translocate hairpin structures? And there's evidence from multiple groups, including Matuszek and Sauer from quite some time ago, that indicates that a number of these AAA ATPases can bind multiple polypeptide chains in their central core. And there's a very nice work recently published from the TANS group showing that ClipB can translocate substrates in a loop conformation. So we wanted to explore this idea, and we wanted to ask a conceptually easier question, which is, can AAA unfoldases bind hairpin structures? So to explore this possibility, we collaborated with Michael Kay and his student James Fulcher, who are very good peptide chemists, and they were able to make two versions of equivalent peptides, 30 amino acid peptides. They were either linear or had their ends joined in a peptide bond, were circular. And Hahn took these peptides and did binding experiments and found that the circular and linear versions of the same peptide bound with almost the same affinity. So this was rather curious. So Hahn determined structures of two of these. And you can sort of see that they look familiar. They look very much like the structure determined earlier with a single chain polypeptide. But on the right-hand side, you can see that now there are two polypeptide chains visible in the central pore of the enzyme. And so what must be going on is, I think, is illustrated here. I'm showing just the Pore loop residues now. And we can only see the two beta strands in the structure because the ends are presumably flexible, but we think the circular peptides must be binding in this sort of configuration, forming a beta ladder through the middle of the enzyme. So let's take a little more close look at this. In dark green, we have what we call the primary strand, and this has the best density, and we're sort of showing all the different interactions made for that here. And the only real key point is that the interactions of this primary strand are the same as we saw earlier for a single chain binding through the central pore of VPS4. The other strand, the returning strand in light green, which has less good density, is not making any contacts with VPS4 really at all. Because of the beta strand symmetry, it's binding to the primary strand, but it is as far away from VPS4 as is possible while still going through the middle of the enzyme, as if it's a passenger just along for the ride. And we can sort of see that in this oblique view here, where you see how it's possible because the transitioning subunit in purple here is shifted a long way from the middle from the hexameric axis. Easily allows a combination of the second beta strand. So one possibility is this sort of gives a model for how AAA unfoldases may be able to start translocation from an internal loop like this. That's one possible mechanism that might exploit this possibility. Another possibility is that this may be the mechanism that underlies how AAA ATPases like CDC48 can translocate ubiquitinated substrates, sort of more complex substrates. Okay, and on that note, I wanted to say, I'll do this quickly. I want to give just a few words about work that we did in collaboration with Ian Cooney and Peter Shen on CDC48. This is a different sort of AAA ATPase. It's each of the subunits has two ATPase cassettes in the one polypeptide chain. There's a D1 and a D2 ATPase cassette that form two rings of the hexameric assembly. And this complex was prepared in Peter's lab by affinity purification on an adapter protein, SHP1, to isolate active CDC48 complexes in the presence of ADP beryllium fluoride. And so the idea was to purify it with an authentic substrate, and that seems to be what happened. There is density for the substrate that goes all the way through the pore. In the C-terminal half, the lower half in this image, you can see the density is relatively good and is reasonably modeled as a beta strand conformation. If we lower the threshold and focus on the first half of the peptide going through the pore, through the D1 ring, it's not so good, but I think this is quite consistent with the model that the peptide is forming a beta strand that goes all the way through both rings of the CDC48 AAA unfoldase. So let's take a look at the binding interactions. We've got the Pore loop 1 residues shown here in the same color scheme as before. There are two positions for the transitioning subunit because focused classification in this case revealed just two positions. But I think this should look quite familiar with a hexameric assembly of the 5 engaged subunits wrapping around the substrate. And it looks very much like 2 VPS4s stacked on top of each other. And we can show that more clearly by doing an actual superposition. So we're superimposing on the ATPase cassettes and displaying Pore loop 1 residues for VPS4 superimposed on each of the 2 CDC48 rings. And the superposition is very similar, implying, I think, that the mechanism is likely to be equivalent. And so let's just illustrate the mechanism in the same manner as before. Now, rather than having one ATP hydrolysis, there's two. There's 2 domains will each hydrolyze ATP. We think it roughly the same time so that they move in coordination with each other. So we're sort of, rather than burning 1 ATP to move 2 amino acids, we're burning 2 ATPs to move 2 amino acids. So we have the potential to do more work, to work on more challenging, more folded and stable substrates. Okay, this is just a final animation representing the overall concept. You have the SHP1 adapter was flag tagged and rapidly affinity purified to bring down an authentic substrate complex that, and we're showing just the linear morphing between the different states that are present in the one asymmetric structure, with the flashes indicating the ATP hydrolysis that will occur in the D1 and the D2 rings. There's a side view, and we'll clarify it further by just removing the subunits closest to the view direction. So I think that idea is pretty clear by now. So I'm going to close by showing you where the work was done. This is a picture was taken a few months ago, and I want to remind you of the things that I've already told you about. So I've talked mostly about VPS4, which is a AAA unfoldase that unfolds ESCRT to drive membrane fission. I've showed you cryo-EM structures of VPS4 and of CDC48 and of structures from other labs of AAA unfoldases in complex with substrate peptides. The binding of ATP at the interfaces stabilizes this beta symmetry of the enzyme, which explains how these enzymes can combine substrates of almost any amino acid sequence. It also implies a hand-over-hand mechanism of translocation. We saw that circular and linear peptides bind in the same manner, and this gives ideas as to how sort of more complex substrates might be translocated. And I'll also emphasize that other AAA unfoldases from our lab that I haven't told you about and from lots of other labs are superimposable. So there's a number of questions for the future. One question is the hand-over-hand mechanism correct? So I think it's very appealing, but that doesn't mean that it's right, and it doesn't have to be right for all triple ATPases or even any of them. And there are competing ideas out there. Bob Sauer, Tanya Baker, and Steve Harrison, for example, had a very interesting structure of ClipX recently, which is a related AAA ATPase. And the structure looks superimposable, but they propose a very different mechanism of translocation. I don't want to describe their model too much, but as I understand that it's sort of going in almost in reverse in a sense, in that well, one thing, it can be stochastic rather than sequential, and rather than having the 5 subunits holding the substrate, the one subunit that moves a long way pulls the substrate over in one go. And so there is, I want to be very clear, not everyone agrees with the mechanism that I've described to you today. And the biggest stumbling block really is understanding of what is the step size. So there's optical tweezers experiments from multiple very good labs that argue that the step size is too big for the mechanism that I described to you. And I think resolving that issue is actually a very important question for the field. There's also a proposal that rather than being sequential, like one subunit at a time, maybe you can form 6 subunits in a helix in one go, and you can fire a whole bunch of ATPase at once and drive a very forceful burst of translocation in a concerted sort of a way. A mechanistic question, which I think is quite challenging, is trying to understand where the ATP hydrolysis takes place. I like the idea that it occurs at the bottom of the helical ring, but the structure at that site, at the resolution we can visualize, is essentially identical to that of the other ATP binding sites. So the question of why hydrolysis occurs there is really a pertinent one. There's a question of directionality as well. So there's good evidence that some triple ATPases can translocate substrates in either direction, from N to C or from C to N. But from a biological rationale, it's clear that VPS4 and some other triple ATPases work in one direction, and one direction will be unproductive and the other will be biologically productive. And so there are still questions of directionality that need to be resolved. I think there's also questions of substrate engagement. Okay, so I'm going to really close now by showing you what the University of Utah will look like in a few months' time, and thanking the people who did the work. I've already mentioned these explicitly for those who played the major roles. So I'm just going to close by saying this is a difficult time that we're facing at the moment in lots of ways, and I hope that you and your loved ones are all safe and well. And that you're, and also we're reminded with the events that are going on in the world that the world is not all a great place, that there are real issues that should be addressed. And I hope that everyone is doing their part to try and both do great science and have fun, and also to make the world a better place for all. And with that, I'll close by thanking you for your attention.
Great, thank you very much. And thank you for a great talk. Fantastic movies. I mean, they came across pretty well through the Zoom, but you know, the molecular mechanism, you know, I'm a sucker for the molecular mechanism movies, I have to admit, but they're really good. So we have a bunch of questions, and we can just go ahead and unmute people and ask. I'll try to go through basically in the order that people sort of submitted them. So the first one was from Svetla McPhee. So I'm unmuting you. Let's see, I think it works. Go to.
Hi, my question is kind of for the early part of the talk. It was not related to bottom. I was just wondered about what defines the 4 nanometer inner diameter of the lipid nanotube. This is really a big stress. The diameter is so too small, but what defines that diameter?
Let me make sure I understand the question correctly. What defines what defines the inner diameter of the IST1 Chimp1b tubes? Is that the question?
Yes.
Yeah, it's defined by the proteins themselves. So if the proteins are assembled into the helical filament without any lipid bilayer around, they form the same structure as if it's in the presence of the lipid. So it's inherent to interactions between the protein subunits.
Yes, I understand. Thank you.
Right, the next question from Sumit Bendikar. Sumit, you can unmute, go right ahead.
Hi, Chris. Great talk. I was curious about the bacterial protein that you use to force hexamerization, and I was wondering if you could elaborate on the rationale for choosing that protein. Were others tried? Are there any endogenous protein binding partners that normally force hexamerization? Thank you.
Yeah, it's a very good question. It was HCP1 was not the only protein that we tried. So a search was done of the PDB for proteins that were stable hexamers that were, smallish and looked like they would express well, and seemed to have termini at about what we thought would be the right sort of position to be able to interact with the C-terminus of VPS4. And so several proteins were selected and tried. And then there was the question of deciding what linker to use, because you can envision that if the linker was too tight, the VPS4 wouldn't be able to assemble correctly. But you also didn't want it to be too long. And that could make the cryo-EM hard and negate some of the advantage of the co-localization. So what Nicole did was to express variants with multiple different length linkers. She varied it systematically in 3 amino acid increments, and each time she assayed the fusion protein by putting over size exclusion chromatography, and that was quite informative. If the linker was too short, actually the complex ran as if it was large, because presumably because the VPS4 subunits couldn't associate when actually being forced to splay out a bit. And so that was an indication of overall conformation. And then it was also assayed for ATPase activity, and it was also assayed for peptide binding. And it was only when the linker was varied until the optimal position was found to be no longer than necessary, but retaining full activity by those metrics. Does that answer the question?
Oh yeah, thank you, Chris. That answers my question. That's really interesting how much optimization there was required. Thanks.
It was a hell of a project.
We have a question about specificity from Jacob Anderson. Jacob, are you there? Mike's not working, so I can go ahead and read it. If the pore pockets are able to accommodate charged hydrophobic residues, linear or circular peptides, how is specificity generated? So, and as an extension, could one predict substrates based on AAA ATPase sequences? And
that's a really good question. And so there's two parts to it. One is the specificity of engagement in the central pore. So let's talk about that. First of all, I should be clear, the resolution is not super high. So I don't want to get too dogmatic about the very precise interactions that are stabilizing the complex. You can sort of see in a general sense, but it's not super high resolution. You can see from the structure that you'd expect different sorts of amino acids to bind with different sorts of affinity. One thing you'd expect is that polyproline would not favor binding because polyproline cannot form a beta strand. And indeed that's true. So we've done quite a few binding experiments that are not published yet, but we find that polyproline doesn't bind. You'd also envision that glycine wouldn't bind very well because side chains seem to be important. And indeed, that's the case. Polyglycine doesn't bind either. And so there definitely are preferences for amino acids for binding, but it's fairly broad. I mean, we do see a range of binding affinities, but it's fairly broad specificity. And this is attractive because there are 12 different ESCRT3s in human, and the leading idea is that all or the majority of the sequence gets translocated and engaged. So there's a need to be able to engage with a wide diversity of amino acid sequences. So the specificity is, in the case of VPS4, and I suspect for many other AAA ATPases, does not come from binding in the central pore. That's fashioned to be a fairly generic binding module. The specificity, I think, will generally come from engagement with the substrate. And I emphasized early on that VPS4 is on its own biochemically only a very weak hexamer. So I think the reason it's recruited to ESCRT3 filaments is because the ESCRT3 filaments display many MIM motifs, and so you have avidity effects by binding subunits to high local concentration with each other. Then they are able to hexamerize, and I think that may be a recurring theme for some AAA unfoldases. But there are other mechanisms as well. So I think substrate specificity may in general be defined outside of the central pore. Does that make sense?
Yeah, I think that does make sense from the mechanism you showed. That you'd have sort of two parts: the recognition part and then the translocation function. And so you can do the recognition up front. We did have a question from Zaid Ibrahim. So Zaid, go ahead.
Hello, thank you for this nice talk. Actually, I have two questions. Maybe there is something I missed in the beginning of your talk. So in your structure, you have the 6 ATP binding sites occupied at the same time, but only one of them hydrolyzed to ADP, right? And that's how it can coordinate the unfolding and the translocation. So only one ATP is hydrolyzed at a time. And my second question is that these AAA plus unfoldase are hexamer that bind to heptameric core particles. And so how can a hexamer bind a heptamer? Is it, are the 6 subunits at the same time binding on the heptameric core particle, or one subunit at a time? And is the unfolding and translocation and degradation happen at the same time, or is this coordinated by ATP as well, the binding to the core particle, or no?
Really good questions. I heard 2 questions there. Let me see if I can take them in turn. So first, I'll start with your second question, which is the 6s under 7 binding mismatch, which is really curious. And so this occurs in the proteasome, where it occurs in ClipX, ClipP, and and this is explained very, very nicely by work from multiple labs now on the 26S proteasome, and has been explained very nicely by 3 groups now on ClipXP, and I would recommend that you take a look at those questions also with ClipP complexes. So how does it happen? Well, there are structures that show that it happens. That and it's a bit complicated in the case of the proteasome because it's the heptamer is a heteroheptamer and the hexamer is a heterohexamer. So I won't say more about it than that. In the terms of the clip X and clip A with clip P, you've got these loops that come down. This is work from other labs. That you've got loops that come down from the clip X or the clip A that have inherent flexibility. And so they have like a binding motif that is able to engage with the binding pocket. But the way in which it reaches it can be different in each of the different cases. And so there's sort of inherent flexibility in the connection. And then 6 of the binding pockets are occupied and 1 of them remains open. I hope that's a clear enough explanation of how it occurs. And then the question of the ATP hydrolysis. The ATP hydrolysis does not seem to be linked in a very tight way to proteolysis. It seems to be really linked to translocation. And the proteolysis just occurs to a first approximation when the substrate gets to the active site. The question of your first question, which I think was the question of the timing of ATP hydrolysis, is really a curious one. So why, if the model we're proposing is correct, why does ATP hydrolysis happen at that lowest site, but doesn't happen at the other sites? We have some ideas, but we really don't know the answer to that. And that's an important mechanistic question, if indeed the model we're proposing is correct.
Okay. Thank you.
Great. So next up, Carlos Escalante had 2 questions.
Yeah, hi. The 2 questions might be related. Nice talk. One of them is that you show in your cryo-EM map that one of the subunits is kind of partially, the density is partially seen. And I wonder what is the significance of that in terms of the function? And also, what is the importance of the hexameric complex being so dynamic that in solution you said they don't form hexamer, right? Other than the being able to assemble around a substrate. So why is not a stable hexamer?
Why isn't it just? Let me see. Again, I heard a number of questions there, very important questions. And if I forget any, please call me out on it. So the first part was we have one subunit which has very poor density. And the way in which that happens is that that subunit is occupying multiple positions when Han and Peter do focused classification on the various VPS4 data sets that we have. I forget, they have something like 12 different positions for that sort of transitioning subunit, and they're all at low resolution, but they're all like at different stages along the same hypothetical pathway of going from one end of the helix to the other. So I just think that's there's very low energy difference between those different states, and so they all exist with some frequency in the preparation. So that's the sort of a technical explanation for it. And then you were saying, why doesn't it form a stable hexamer? Well, some of them do. So VPS4 does not, but CDC48 does. And there's a range of these, and I think they may be related to the different ways in which they engage with substrate. With VPS4, it's attractive to think that, in a sense, this is a very dangerous activity to have, right? Because once any substrate gets to the pore, it can be unfolded. And you don't want these machines going around just unfolding every protein in the cell. So you have to have a very regulated mechanism to control who can get to the active site. And one way of doing that is to make sure that you only assemble the active form at the right place, such as when you get avidity interactions formed by the polymer that you subsequently need to do action on, which I think is what's happening with VPS4. But with CDC48, it's a different mechanism, it seems, entirely, because there are multiple adapter proteins which can bind to the N-terminal domains of CDC48 that I didn't say much about, that can deliver specific substrates, sometimes in response to ubiquitination. And so enzyme with substrate, and hexamerization is, I think, one of them. Does that answer the questions?
Yeah, thanks.
Great. Well, with that, I think we can wrap up. Chris, thank you very much for a great talk, and thank you for joining us. And everyone, Chris's talk is recorded and will be posted on the SBGrid TV channel. There's a lot of other talks and content over there. So if you missed any of our previous seminars, you can check them out over there. And everyone, stay healthy, stay safe. Thank you very much.
Thank you. Bye-bye.